Thank you, Nathan.
Great. Thank you very much. This is John Hare, and I work for the Northeast Fisheries Science Center out of the Narragansett Laboratory, and I have the pleasure to introduce Nathan Rebuck. He's a National Research Council postdoc who's been working at the lab now for about 2.5 years. And the work that he's going to present today is a lot of what his postdoctoral work is. Nathan got his bachelor's at Princeton University and then went up to University of Maine and got a PhD with Dave Townsend. And currently he's working with developing an ocean acidification climatology for the region, and that's the work he's going to talk about today. And he's also working with other scientists at the center on chlorophyll climatologies, and he's continuing to work on his nutrient climatology. So without any further introduction, I'll pass it over to Nathan.
All right. Thanks, John. What we'll be talking about here today, kind of an overview of the shipboard surveys that have occurred on the U.S. Northeast Shelf, and really want to talk about quantification of some of the variability in the carbonate chemistry here in our region and some of the potential biological implications. And here's my name and an email address in case you have any further questions after the talk that don't get answered. So to run through, give a quick ocean acidification primer. Hopefully, just by virtue of the fact that you've tuned into a talk about ocean acidification, you'll be pretty well caught up on what exactly that is. And then we'll get into some analysis of the spatial and seasonal variability. Look and see if we can find a trend over the last 30 years from in situ measurements and compare the scales of some of the variability that we're seeing here on the coast, and then get into some of the factors that need to be considered when we start talking about translating the knowledge of acidification of the waters to management and potential impacts on resource species. And then we'll just close up with a summary synthesis at the end. So, as everybody hopefully knows, ocean acidification has been called the other CO2 problem, typically conceptualized as a change in ocean pH that results from increased atmospheric carbon dioxide concentrations. But in addition, it also affects the partial pressure of carbon dioxide in the water itself, the amount of dissolved inorganic carbon in the water, and the degree to which this acidification occurs can be moderated to some extent by the alkalinity in the water. And this is a slide I'm sure most people have seen before. It's the famous Keeling curve, and this was taken from Feely et al. In 2008. But it just shows that as atmospheric carbon dioxide increases, so does the partial pressure of dissolved carbon dioxide in the water. And as you can see in the blue, it's much more variable than the atmospheric carbon dioxide. And in the meantime, excuse me, there's also been a decrease in the pH of the waters out there in Hawaii as well. And over the last decade, this record shows that there's been about a 0.02 decrease in pH. And the reason that carbon dioxide acidifies the waters can be shown here in this little schematic, that the carbon dioxide in the atmosphere enters the water, becomes the dissolved phase, and then it dissociates into, up here at the equation on the top shows the carbonic acid, bicarbonate ions, and then the carbonate ions. So just kind of a brief overview of the entire system. pH is a measure of the hydrogen ions that are floating around in the water. It's affected by both the carbon that's been, the carbon that dissociates into the waters, in addition to a variety of other ions that are floating around and their activities. But it's pretty sensitive to errors, and historically there's a pretty pretty disturbing lack of precision in a lot of pH measurements, which doesn't necessarily mean that their historical measurements aren't valuable, and hopefully I'll convince you of that here in a little bit. But it's fairly intuitively pleasing, and a lot of people have familiarity with it, not just marine scientists, but other scientists and people in the population. And pH can be run on a continuous system, like Rick Wanikoff talked about last week. You can just slap the equipment into a ship, and as it tools around, it can measure pH with relatively little influence from people. Alkalinity, it's a little more time-consuming to measure. It has to be done on discrete samples that are collected on board a ship or I guess, from the water's edge, but then it needs to be returned to the laboratory to be run and analyzed. The important thing with alkalinity is that there's no change in alkalinity with increasing atmospheric carbon dioxide directly. So what I use it for here in a lot of this presentation, when looking at some of these decadal scale trends, it should serve as a little bit of a control to determine whether or not your data have any significant errors in them. Also, dissolved inorganic carbon, again, it requires discrete laboratory analysis, and the machinery requires frequent calibration. But the sensitivity of measuring dissolved inorganic carbon is a little bit higher than measuring the pH. It's not subject to some of the noise and large swings that the instruments can provide. And then also the partial pressure of carbon dioxide, PCO2, that can be run autonomously and continuously on a flow-through system. And again, Rick Wanikoff mentioned this last week, getting some really good data sets through some ships that are outfitted with these sensors out there on the Northeast Shelf. But the important thing is that with any two of these measurements, you can calculate all four of them in addition to the saturation states. So what is the saturation state? Well, the saturation state is the thermodynamic stability of calcium carbonate in this instance. There's two forms of calcium carbonate that we're interested in, calcite and aragonite, and they have slightly different dissociation constants. But the one that most people are interested in is aragonite because that's used by most of the calcifiers in the marine world. But the calcium carbonate, basically, the saturation state describes whether or not the degree to which the water itself is corrosive to the solid form of calcium carbonate. And values at 1 are just at saturation. Anything less than 1, the water actually becomes highly corrosive. Anything above 1 is thermodynamically stable in the water. But that doesn't mean just because you're above saturation state of 1, all effects on biology are equal. Anytime you have a decrease in saturation state, you're going to be stressing some of the organisms. And pelagic systems, like I have here, are typically around 4 to 6 in saturation state. Here on the coastal shelf, we're usually between about 1 and 4. And the saturation states are kind of becoming used to express the full suite of changes throughout the carbonate system. You know, it's kind of clunky to continually talk about 2 or more parameters when you're trying to describe acidification, and it seems like the community is moving towards using saturation state as a single scalar variable that we can use to quantify all of the changes. And just to mention, sort of counterintuitively, an increase in dissolved carbon dioxide will lead to a decrease in carbonate ions and actually decrease the saturation state as well. And like I mentioned, alkalinity can be thought of as the buffering capacity of the water. And for stable source waters, alkalinity really tracks as a function of salinity. This is a figure I borrowed from Alec Wang in his recent paper that shows a pretty darn tight linear relationship between salinity and alkalinity. And these two circles up here, this is Gulf Stream water, source water, and this is Labrador sea salt source water. And there's pretty conservative mixing in between the two. And the normalized difference, if you had those two source waters at the same salinity, would only equal about the same difference as a salinity difference of 1 unit. And where we're working on the shelf, our salinities are usually somewhere down here in the 31, 32, up to 34 range. So really, the changes in salinity have a pretty strong impact on the alkalinity found in our waters. So kind of to put a little bow on this, the magnitude of ocean acidification going forward will be determined by both the global increases in atmospheric carbon dioxide and the local salinity that's found in the water itself. And these changes will also be occurring on top of the natural background fluctuations of abiotic temperature and salinity cycles that are occurring out there, phytoplankton bloom uptake of carbon dioxide, and then the subsequent respiration of that productivity, and also the regional scale hydrographic variability, such as changing source waters in the Gulf of Maine from the Labrador slope waters to the warm slope waters down below. And of course, the acidification will also have biological consequences. What these are, it's tough to say. There have been demonstrated effects, but they're inconsistent across similar species. But things have been shown to harmfully affect, acidification has been shown to harmfully affect corals, certain pteropods and coccoliths, a lot of bivalves. Some crustaceans actually seem to grow heavier body mass. But there's also been effects shown on ichthyplankton and fish eggs, larval stages, and also on elemental biogeochemical cycling, such as nitrification and nitrate fixation. But the important thing is that we need to know the mechanism of how this suite that we call acidification is affecting these species. So for example, a lot of the work on finfish has really focused on how the pH can generate stress on some acid-base reactions and certain ion transport in larval fishes, whereas more shellfish are probably concerned, well, they're not concerned. We're concerned with the way that saturation states decrease the growth due to their inability or a decreased ability to lay down the calcium carbonate to generate their shells, whereas other studies have shown that it's actually just a direct amount of dissolved carbon dioxide that has increased photosynthetic uptake and spurred extra productivity in some waters. So getting on to the data that we'll be looking at, this is examining the variability in a multi-year data set. To look at the data, the first thing we did was fit a first-order harmonic to the seasonal cycle for all of the carbonate parameters on the northeast shelf. And then we spatially interpolated the seasonal residuals and averaged all the years from our data to produce one climatology. And then third, we compared this climatology to more recent data to look for some decadal trends. And the data we're using is from the Marine Resource Monitoring Assessment and Prediction Program. We call it MARMAP. It was run from 1976 to 1983 by NOAA and the Northeast Fisheries Science Center here. The project was designed to look at phytoplankton primary productivity, so it's only been in the euphotic zone. So all the data that I'll be showing here today is just for surface waters. I don't have any data at depth. The methodology precision comes down to about 0.02 for pH and about 12 micromoles per kilogram for alkalinity. These error bars are about 10 times larger than the contemporary methods, but again, it doesn't make them worthless. It just means you have to match the scale of the feature you're examining to the scale of error in your data. And all the climatologies I'll be showing, I've normalized the PCO2 levels to 2010, basically tacking on 1.58 parts per million per year since the date of collection. That way the climatology can be compared directly to new data. Of course, when we're doing the decadal examination, I don't normalize the data to any years. But these figures here on the right show the data distribution for this program from 1976 to 1983. As you can see, there's pretty good spatial coverage, especially in the spring and summer, and they range from the right off Cape Hatteras all the way up into the Gulf of Maine and the Bay of Fundy. And on the upper left-hand side, these are the 4 regions that I've divided into to calculate the seasonal cycles. So we've got the Gulf of Maine, offshore Georges Bank, southern New England, basically running from about Cape Cod down to Hudson Canyon, and then the Mid-Atlantic Bight, more southerly than that. So to look at the seasonal and regional variability in pH, this is, like I mentioned, the first order harmonic fit to the data. And then in the center, this is the winter average pH. And then on the far right is the summer average pH. And as you can see, there is a thermal effect on the seasonal cycle. The pH is much higher in the winter time and then decreases about the same time that the water starts increasing in temperature. And this is compounded by biological activity through some respiration processes there towards the end of the summer. Also, there's a general decrease in pH from the southwest to the northeast. So as you can see, as you move from the mid-Atlantic up into the Gulf of Maine, there is a decrease of pH. And there's also an inshore-offshore gradient, most notable over here in the summer. As you can see right along the coast, the pH is slightly lower than the offshore regions as well. And I also want to point out that the Gulf of Maine and Georges Bank have the lowest seasonal amplitude here on the left-hand panel. The Gulf of Maine is the dark line that runs through, while Georges Bank is the evenly spaced dashed line. So they don't vary quite as much in terms of pH as the southern New England and mid-Atlantic areas do. Moving on to the aragonite saturation, the seasonal cycle here is opposite of pH, where the lowest aragonite saturation values occur in the winter. The primary drivers for that are likely the total dissolved inorganic carbon, and also there's a slight alkalinity effect as well. Similar to the pH, there's a decrease from southwest to northeast, where the Gulf of Maine is showing lower saturation values than down here along the mid-Atlantic. And also there's a decrease in saturation state in coastal areas near freshwater sources. I don't think there's actually any data within Long Island Sound, but they did get right up to the mouth of Delaware Bay and down here at Chesapeake Bay. So this is a realistic interpretation here out in front of the coast. And all the regions here are roughly similar in amplitude. The Gulf of Maine is a little bit lower in saturation state than the other regions, but for the most part, the seasonal variability is about the same. When we're looking at the PCO2, same general trends. There's an increase from southwest to the northeast in the winter, shown in the middle here. And as you can see, in the summer, we get into some fairly high variability spatially. So we've got these interesting little data points which stand out from the rest of the background. And that just shows that PCO2 really is a fairly noisy measurement. If you're measuring it, you really need a lot of data points to filter out some of that small-scale variability that occurs naturally. And finally, getting along to alkalinity, there is a pretty strong alkalinity seasonal cycle. There isn't one so much on Georges Bank. This is the Georges Bank line up top. And that's because Georges Bank doesn't go through the same salinity cycles that the inshore Gulf of Maine or the mid-Atlantic does. And this is driven almost entirely by the salinity cycle. And again, I just want to point out that there are important differences in the seasonal cycles between pH and aragonite saturation. So, like I mentioned, if you're worried about low pH waters, you're probably going to be focusing on things in late summer and things that are down along the mid-Atlantic bight or here in southern New England. Whereas if you're thinking about low saturation states, you'll probably be more focused on winter waters up here in the Gulf of Maine. So depending on what the potential mechanism that disrupts the biology, you really need to take into consideration the spatial and seasonal variability of these parameters. So moving along to the decadal scale trends, what I did was take an average of all the MARMAP years from 1976 to 1983, and that generated the 1980 climatology. To compare to that, we have recent data from a joint NOAA-NASA program. It's called the Climate Variability on Primary Productivity and Carbon Distributions in the Mid-Atlantic Bight and the Gulf of Maine, shorten that to CLIVEC. And we have 3 cruises for that one in August 2009, November 2009, and November 2010. And the differences, as calculated, the images just show the bulk 30-year change, and then they're reported as per decade. And the station locations for 2009 and 2010 are shown on the map. So here's the difference that we've seen here. The difference from 1980 to 2009 in pH, on average, per unit area has been about 0.01 pH units per decade, and the spatial variability in that's about 0.006 is the standard deviation. And in terms of the change in alkalinity, the average change is 5.3 per decade with a standard deviation larger than the average. So like I mentioned, alkalinity shouldn't be changing. I mean, it can because of variations in salinity and varying source waters. But for the most part, the alkalinity should remain about the same from 1980 to 2010. And I think by virtue of the high variability, standard deviation of the spatial variability being greater than the overall average, I think that gives credence to the fact that things aren't changing very much. In addition to the fact that these values should be around 2200, so it's about a 0.1% change. Whereas the pH, it's 0.01 per decade, and this is over 3 decades. So the average change is about 0.03, and like I mentioned, our measurement error is about 0.02 for the 1980s data. So it is larger than our measurement area, and most importantly, it's pretty consistent in direction. You know, with the exception of this one data point down here off the Delmarva coast, pretty much everything we're seeing is moving in the same direction. And then when we look at the dissolved carbon dioxide, the PCO2, we see similar pattern. The partial pressure of carbon dioxide is increasing by about 16 parts per million per decade. The standard deviation of 10 is lower than the average for that. And then when we look at the change in aragonite saturation, it's about 0.12 per decade, and the standard deviation is also smaller than that. So there is some spatial variability when comparing these two fields, but overall the trends for the entire Northeast Shelf do seem to be moving in the direction that we'd expect, because we know that atmospheric carbon dioxide concentrations are increasing and the pH in our waters is moving downwards, and it's reflected here in the analytical record. So kind of to put some of this in perspective, I want to show the magnitude of pH variability in marine systems overlaid with the variability that we're finding in our analysis. So this is the pH scale here on the left. Down at 7.8, you could find that in much lower salinity estuaries, very heterotrophic productive waters in a sort of a post-bloom period. Probably wouldn't find it around large areas of the Northeast Shelf, but it could pop up here and there. And algal blooms up around 8.4. I doubt we'd get that high very often, but maybe if you were in a bay undergoing a very strong phytoplankton bloom, it's possible. But the average open ocean is about 8.1, whereas here on the Northeast Shelf, our average in 2010 is about 8.05. And there is some diurnal variability that we need to take into account. It's about 0.05. This has been measured by the PMEL carbon group up at UNH, Joe Salisbury and those guys. Their diurnal variability, it's right off the coast of New Hampshire, so it's actually probably an upper bound estimate, being at the mouth of an estuary instead of being in a more open ocean environment. But that shows about, this red bar shows about the magnitude of the change that we can find diurnally. And then the seasonal variability, the large bar in the middle is the seasonal variability calculated through our climatology. That's about 0.15, whereas also that same buoy up off of New Hampshire has shown that their seasonal variability is closer to 0.25. And I think the primary reason for that is they're catching a lot of the extremes, the extreme events that are going on out there, whether it's the giant rainstorm or peaks in productivity. They're really getting the full range of pH that occurs out there, whereas our average really rounds off those errors to produce the climatology. Since pre-industrial years, which has been about 200 years, Meal et al. Calculated that we've decreased global ocean pH about 0.11 units. And of course, that's a one-way street. So while diurnal and seasonal variability are kind of bouncing around on their appropriate timescales, this has just been a slow one-way downward motion. And on top of that, this is the measured global change since 1980. Like I mentioned, we can detect this in our instrumental record, but as you can see, the variability of the diurnal and seasonal is much larger than what we'd expect over 30-year change, which should come out to about 0.06 since 1980. And then moving on, this is a projection of what pH will be doing in the next 40 years. This was published Orr et al. in 2005, and also some of the more recent Earth system models are coming into agreement with that, showing that in the next 40 years will probably decrease about 0.12 units. And then in the next 90 years, we're also seeing changes on the magnitude about 0.31. So when you put all those together, it really gives a sense that these one-way streets are, the diurnal and seasonal variability will continue to bounce up and down, but this scale of variability will just slowly begin to shift down. And in some sense, we've already moved almost a full seasonal, a half seasonal amplitude lower from pre-industrial times to present times. And then in the next 40 years, we'll probably move another half seasonal amplitude down. So our average will be down here, and then the seasonal variability will be bouncing around in this vicinity. So what are the potential impacts going to be on resource species? Well, I don't have any hard and fast answers to that, but I have some ideas about what's going to be important to take into consideration. And the first thing is the distribution of organisms during various seasons and their spawning characteristics. And also we need to know the mechanism of potential acidification perturbation. And this all just arises because there's a lot of complexity in this system in terms of thermal preference of organisms, depth preference, life history, and also their location. So just to give you an example, these are the trawl survey abundances for longfin squid up in the upper left-hand corner. Excuse me. Shows the distribution of squid in the spring, which is about the March to April time frame. And as you can see, almost all the squid are found on the edge of the shelf break and probably off the shelf where we don't sample. Whereas in the fall, the squid come back up onto the shelf and flood the inshore waters all the way from Hatteras up into Massachusetts Bay and onto Georgia's Bank. This is important because they spawn year-round, and they encounter pH conditions which are very different out on the shelf versus up in the coastal areas. So while they're out at the edge of the shelf in the winter and spring, they're in a very high pH environment. Seasonally, they're in the highest time of the year, whereas once they move inshore in the fall, they're getting into lower pH values both spatially and seasonally as the seasonal pH cycle swings down. And pH has been shown to have an effect on the larval stages of the squid. Kaplan et al. In 2013 just put out a paper. So it'll be interesting to see whether or not the changes cancel each other out, whether since the squid spawn year-round, if the spring distributions remain unchanged and recruitment goes on as normal, whereas the fall distributions might be affected to a greater degree by the decrease in pH due to their movements on and off the shelf. So just as another example, American lobster, we usually don't think lobsters would move too much, but here in the spring we find them in deeper waters here in the Northeast Channel and really across the coast of Maine, whereas In the fall, we find them up on George's Bank and a little bit further down here off of Rhode Island, Martha's Vineyard. And for lobsters and other crustaceans, we're probably more concerned with the saturation states as opposed to direct pH effects. And what we see is that the lobsters will be encountering up in Maine a very low saturation state relative to those that are out at the edge of the shelf. And particularly if they're spawning here in the springtime, they're going to encounter a very different saturation state of 1.6 versus later in the fall where it's up around 2.8. So you really need to take into consideration what the mechanism is going to be that's going to influence organisms. It can't just be the suite of changes encompassed by ocean acidification. And in moving forward, we're hoping to incorporate some projections of carbonate chemistry to these climatologies to really downscale, to tease out what some of the changes will be on this regional perspective instead of just looking at a global perspective like those models currently do. We'd like to continually monitor in situ conditions through NOAA and ship of opportunity sampling. Try to get these included into the Northeast Fishery Center Ecosystem Status Report. We also would like to integrate the climatology products with other regional partners to give a better description of the overall carbonate environment. And ideally, we need to get these products of climatologies and future projections to population and ecosystem modelers to see how these changes are going to play out when it comes to resource species and fisheries recruitment. So in summary, I think the most important thing is that we need to recognize that there is a large natural variability, both spatially and seasonally, of the relevant ocean acidification parameters on the Northeast Shelf. The decadal scale acidification, as measured by in situ measurements, is near the published global averages, but making direct comparisons can be complicated by the spatial variability and historical measurement precision. The seasonal extremes of pH and aragonite saturation are out of phase and geographically separate. Therefore, the projected impacts on resource species have to include a mechanism, that's not just acidification descending as a blanket evenly across the region and across the year. And finally, exposure to acidification will depend on an organism's thermal preference, where in the water column it resides, seasonal migration patterns, and its reproductive history. Consequently, certain populations might have a naturally high acidification tolerance, like those in estuaries or even diadromous species, whereas a lot of others might not, such as pelagic or offshore benthic species. And with that, I'd just like to acknowledge some of our data sharing partners, Rick Wanikoff at AOML, Joe Salisbury, Doug Vandemark at UNH, Alec Lang at NOAA, and I'd like to thank NOAA, National Research Council, for funding and opportunities.